Assalamualaikum and hi everyone. My name is Saida binti Sabran and my meeting number is 210425. So today, my group will be presenting one of the biochemical tests which we have learned throughout this course, which is centrifugation in blood sample analysis. This is the outline for our presentation today. So we first start with the introduction, second, significance, third, method, fourth, mechanism. Centrifugation is a technique used for the separation of particles from a solution. It is influenced by the density, temperature, distance of particle displacement, and rotation speed. Each centrifugal machine has its own use and function. Analysis of blood samples requires centrifugation prior to the analysis to separate blood cells and other solid components such as paper from serum or plasma. It is important as it helps to diagnose diseases and condition and find out how well our organs work.
Explain further for the next part, which is significance of centrifugation of a blood sample. The significance of centrifugation of blood sample are very broad and various, especially in clinical and laboratory sectors. One of the significances is centrifugation enables the separation of blood cells and other solid components such as fibrin from serum or plasma. It is important to separate the cellular and liquid portions of a blood specimen as soon as possible when the test requires a sample of serum or plasma. The blood bank utilizes centrifugation process for antibody screening, cell washings, and tube typing. Centrifuges are also used in coagulation department to obtain platelet for plasma. The second significance is centrifugation of blood sample assists in disease diagnostics and research basis. As explained previously, the separation of blood components helps in diagnosis of disease, as some of the blood components can be used to detect infectious diseases. Other than that, some components of the blood such as leukocytes can be used to study immunological functions such as cytokine production and surface marker expression. Last but not least, other significance of blood sample centrifugation is it helps in measuring the volume fraction of erythrocytes in blood. To assess anemia and reduce oxygen carrying capacity, a measurement of hematocrit or hemoglobin is performed. Whether hemoglobin or hematocrit is preferred depends on different needs and often determined by tradition.
Hi everyone, my name is Nazir Amin and my metric number is 212624. In this slide, I will talk about method for blood sample. The method that we use in the blood sample is plasmapheresis. Plasmapheresis is to spread the blood between blood plasma, white blood cell, platelet, and red blood cell. The type of centrifuge used is microcentrifuge, or we call it table-stop centrifuge. Type of rotor that are used are swinging out rotor and fixed angle rotor. The time that for the separation of the blood around 50 minutes, and the speed that I use 3000 rpm. For the volume of the tube, we are using 1.5 ml until 15 ml. That's all from me. Thank you.
I'm Hemla Anak Perempuan Chandrin with the metric number of 210011, and today I'll be presenting part of the mechanism. So, centrifugal force is the rotational force acting outwards on object from the center point. As you now know, centrifuge is a machine through which centrifugal force is applied to substances that one wishes to separate. For instance, is the separation of blood sample into blood cells, platelets, and plasma. These three substances of different densities together make up our blood. When placed into a centrifuge and acted upon by a centrifugal force, blood cells, platelets, and plasma will separate. Within a solution, gravitational force will cause particles of higher density than the solvent which is the red blood cells, to sink, and those less dense than the solvent to float to the top. And next up, Litya will be further explaining about the mechanism of centrifugation in blood analysis.
Good morning, I am Litya Subramania, metric number 212571. Now I would like to continue explaining on the mechanism of centrifugation. As previously explained by Hemla, gravitational force will cause particles in a solution that have a greater density than the solvent to sink and particles that have a lower density than the solvent to float to the top, which causes platelet to float above red blood cells. In this case, because the white blood cells and plasma are even less dense than the platelets, they will float above the platelet layer after centrifugation. Centrifugation takes advantage of even minute differences in density to separate particles within a solution. Nextly, as the rotor spins around a central axis, it generates a centrifugal force acting to move particles away from the axis of rotation. During centrifugation, heavier particles rotate at a longer radius and lighter particles rotate at a shorter radius. If the centrifugal force exceeds the buoyant force of liquid media and the frictional force created by the particles will sediment. This figure here illustrates the relationship between the centrifugal force and buoyancy friction within a solution.
Before we end our presentation, I would like to recap the things that we have presented. First, we have learned the concept and the practical use of centrifugation. Next, we have looked at the method on how the blood analysis will be done through centrifugation, which is plasmapheresis. Third, the mechanism and the purpose of these biochemical tests have been understood.